You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Richard Jacobs with the Future Tech and Finding Genius podcast. I have Priya Nimish Deo. She's at the Department of Oral Pathology and Microbiology, Bharati Vidyapeeth University, Dental College and Hospital in Pune, Maharashtra, India. So Priya, thank you for coming. How are you doing?
I'm good. Thank you. I would like to thank you first for the invitation to share my views on this interesting topic.
Work on the oral microbiome. What is interesting to you about it?
The very interesting thing at the outset is that currently microbiome is linked to many different diseases in the human body, right from Salmonella typhi to the gallbladder cancer and H. pylori to the gastric cancer and so on. So that is why there is a huge interest among the researchers to study the role of oral microbiome and in oral diseases that range from caries to oral cancer.
That's microbes. I can understand that microbes in the mouth can cause cancer or disease in the mouth. But how could microbes in the mouth cause gallbladder cancer, for instance? How do you think that is happening?
No, I was just giving you an example.
I have read that microbes that are usually in the mouth can appear in blood vessels or can appear in other parts of the body. I guess they migrate out of the mouth through the bloodstream.
Yeah.
So what in particular are you studying about the oral microbiome?
I would like to study, like I have put up a review articles which is describing the role of oral microbiome first in health. What role does it play in health? And then it would be very interesting to study what role the oral microbiome is playing in the etiology of oral precancer and cancer. When I say oral precancer, it means the oral potentially malignant disorders, meaning which have the potential to transform into malignancy if left untreated. So that would be interesting to study the role of this microbes in oral cancer, oral precancer.
Are you doing experiments right now, or are you not at that stage yet?
Not at that stage yet, but yes, would like to do in the future.
You've been studying the oral microbiome. Have you done any experiments yet under another professor?
Not as yet. I am at the stage of collecting data and different things and just reviewing the existing literature on the oral microbiome.
What have you found that is very interesting to you in the existing literature? Anything very interesting?
Yeah. I would like to say that previously when the microorganisms could be identified only by culture methods, means only those microorganisms which could be cultured could be identified. But with the advancements in technology and with the uh, upcoming of technologies like next generation sequencing and different sequencing technologies, it has become possible to identify also those microorganisms which cannot be cultured. So there are around 700 different species of microorganisms have been identified in the oral cavity. With the help of sequencing technologies, it has become possible to identify even the microorganisms which were not identified till today. Hence, it has become a very interesting field.
Why can some microbes not be cultured? What makes it hard to do it?
Because they do not have that environment which is required for culturing them in a laboratory. That is why some organisms will directly be sequenced, and if there is something called as the human oral microbiome database which is available, and if you can sequence your sample and you find a match by using the reference in this database, then you identify that particular organism. It goes like in that way.
Okay. And I know that there is 16S sequencing,
Yes,
but there is
yes,
also whole genome sequencing
yes,
now
yes,
of the
yes,
microbes, right?
yes.
And there is also metabolomics, where you can look at the metabolites.
Metabolites produced by these microorganisms.
What other kinds of sequencing can be done?
There are different
Proteomics?
there are different kinds of sequencing which can be done. Like first in the basic is the 16s rRNA or rDNA sequencing, wherein the it sequences only that particular genetic marker which is specific for bacteria. And there are other omics technologies like proteomics, genomics, metabolomics, and metagenomics, which identify, like for example, metagenomics will deal with not only who are present but it will also identify what is their functional activity. Like not only it will not only help in observational study, but it will also help in what is going on or what are the microbes actually doing in the body. That will be able to be studied by metagenomics. Proteomics will help in the identification or studying of the proteins produced by these microorganisms. And metabolomics will identify the different metabolites of these produced by these organisms. So these are the different omics technologies for microbiome research.
Okay. And have you seen anything in the literature where someone has sampled every possible way the oral microbiome? Have you seen anyone that has done sequencing every way
Yes,
that they can of the same sample?
of the same sample, meaning of the oral
Yeah,
sample.
have they looked at a particular subject and done every kind of omics you can do on that one
On
person?
the same sample, every kind of
Yes.
omic. I have not come across any article wherein on every sample the different omics is done.
I know it would be expensive and difficult,
Yes, yes. Still,
but
the cost of this sequencing is actually it is decreasing day by day, but still on a higher side.
so what are some interesting things you have seen when they use transcriptomics or they use metabolomics or metagenomics? What kind of interesting things have you learned?
So I am actually I've still not gone into the experimentation. So my main thing would be only in the review part. Like it is like microbiome as it responds to everything. It changes in the diet, your changes in the lifestyle, your changes, your disease state, your systemic condition. So the microbiome varies with each and every change which occurs in your body. So that would be an interesting part to study. And this study process also will vary depending on the standardization of the methods which are involved, and the variability in the results can stem from any steps, right from the sample collection to the DNA extraction to sequencing.
Um, how fast do the bacteria in your mouth divide? Do you know what the average is?
They are actually, I don't know the exact numbers, but they are actually continuously in a state of something called a cell cycle, where they continuously are moving from one stage of the cell cycle to the another, and continuously dividing.
On average, do you think they're dividing very slowly or quickly if you compare them to the gut?
They can double within 20 minutes, 20 to 30 minutes.
Oh, I had thought in the mouth that they, for some reason, that they divide slower. So within 20
Slower
to
than
30
the gut.
minutes, okay, so within 20 to 30 minutes, they can double in the mouth.
Yeah,
Wow,
they
that's very
keep on
fast.
multiplying, and they at the same time as the number also increases or decreases depending on all the other environmental factors also.
Do you think that when you're not eating, the bacteria would divide slower, and when you eat food, that they would suddenly divide much faster?
Maybe.
Any other conditions under which you think they would suddenly become very, very active?
In case of whenever there is some kind of infection or any systemic disease, it is many a times first reflected in the mouth, the oral cavity. At that time, then the multiplication of these bacteria would definitely be faster than when a person is much more healthier.
Oh, just from infection, or when they're going into a precancerous or a cancerous state?
Yes, at that time, definitely. The cells would be multiplying more faster than when they are in a healthier state. In any infection or any diseased condition, that includes cancer also.
So, in the literature, are there many studies on the metabolomics of the mouth or the proteomics? Have you seen any interesting
Actually,
studies?
there are interesting studies, but still, as I said, this microbiome research is at a very younger stage or a primitive stage of development, and still large scale studies are going on, and data is being continuously added, so that a basic database is created mm, about the microbiome in health and disease, after which only it would be actually be applicable for human treatment.
I've heard that there are many different niches in the mouth, above the tongue, on the cheek.
Yeah.
How many different microenvironments do you think there are? I mean, like, do you think that the microbiome is different for each tooth?
Yes.
Or if a tooth has a
Not each
filling?
tooth. Yeah. Not particularly in each tooth. It will be different for different niches like the tongue, the teeth, the gingiva, the buccal mucosa, the teeth wherein there will be a lot of plaque. So the oral cavity is a highly heterogeneous ecological system containing different distinct niches with significantly different microbial communities in each niche. So the composition of the microbiome will vary from area to area within the oral cavity also.
Any other interesting things that you have learned by reading the literature? Because I've only read a very
That all
small part of it.
that
But what have
yeah
you
that
found?
that there is a myth that all microbiomes are bad. So but there are many useful bacteria in the body which play a very important role in the digestion, in the gut, in maintaining the gut health, and in the functioning of the immune system. So it all depends on the balance between the good and the bad.
Okay. Any other interesting things that you have learned that you didn't know before you read some of the literature?
Like mainly is that oral microbial communities cannot be understood by the isolated of individual microorganisms, but they are much more complex, and it is very important to study them in association with the host. That is very important. How they impact the host and how they stay in harmony within the human body. That is important. So the relationship between these microbial communities and the host is important than to study them isolated or individually.
Do you think that if I have a certain microbe and there are many, many of them in the mouth, that it will produce certain metabolites? But if that bacteria becomes very few in the mouth, that it will produce different metabolites. What I mean is, do you think bacteria produce different metabolites depending on whether they are dominant or not dominant in the mouth, for instance? Do they change their function as they are prevalent?
They may change their function in a disease state. That is something called as dysbiosis. When the balance is disturbed, they may change their function. When the balance is disturbed,
How would you know if they changed their function? How would a scientist figure that out? Would you look at their metabolites, or would you look to see if their gene expression had changed, or their protein expression had changed?
yes, all they expressed and the sequence that might help in understanding whether the what role it plays. That is only the uh, branch which is called as metagenomics, which will help understanding the functional activities, what is going on in the body, what the bacteria are actually doing in the body, which will help shifting the researchers studying from observational to functional studies.
What do you think the role of the bacteria in the mouth are? What is their
Yeah.
job? Why are they there? Are they there just to feed off of us? Or what useful functions do you think that they provide in the mouth? Like bacteria on my tongue, what function do they provide that's beneficial to me? Do they make certain compounds? Like why are they
They
there?
promote
Or are they just getting free
yeah
food?
yeah they play a crucial role in maintaining oral homeostasis by protecting the oral cavity and preventing disease development. They are present in the human body, they and playing a critical role in physiologic, metabolic, and also immunological functions. All these functions include many, like digestion of food. They help in the digestion of food, nutrition, differentiation, and maturation of the host mucosa, the immune system, the metabolic regulation, the barrier function of the skin and the mucosa, and the balance between pro-inflammatory and the anti-inflammatory processes. All this is where they play a role in the different microbial communities by interacting with the host.
Well, have people been studied that have taken a course of antibiotics? And what happens to their mouth when they take antibiotics? Have they been studied after that to see what happens, or during it? What happens to their mouth bacteria? It supposedly would kill all of them. New ones would come. But what if you sampled someone before they took antibiotics and while they were taking antibiotics and just after? What would that look like?
Antibiotics are powerful medicines which will kill the bacteria and will stop the bacteria from reproducing, and they will destroy them.
What data are you gathering right now? Are you starting experiments, or what stage are you at?
I would be wanting to work on the different kind, what role this microbiome is playing in disease. Yes. I
So are
would
you
be
working with
wanting to
a
work on that.
professor, or
Yes,
what is your role at the okay? So when will you have your data finished? When will you be able to write your first paper?
this was my first paper on the review of literature on the oral
Oh, okay.
microbiome in health. So after that, I would like to write something on oral microbiome in disease. uh, In disease,
Can you
particularly cancer, but in disease in general.
is your
That
paper in English and in Hindi, or
no,
what
not
language is it in?
in English only.
Okay. Do you think that for listeners you can provide a copy of the paper so we could attach it and read it. Would that be okay?
The one which is already published, right?
Yes.
Yes, sure, definitely. That is already online.
Well, sometimes it's hard for people to access.
Find
So
out.
if you have
Okay.
if you have a copy of it,
Yeah,
that would be
should
good.
I
If you're
mail
willing
it to
to
you?
share, if you have a PDF, that would be good.
Yes, I have a PDF.
Okay. Yes, please. Well, very good, Priya. How long do you think it will be until you do your next paper? 6 months, a year?
Yeah,
How long do you think
maybe
it will take?
no, 6 months. I've already in the process of completing it.
Okay.
Yeah, so around 6 months it should be published.
Okay, and for people listening, how can they get in contact if they want to ask you questions or find out more?
Could I share my email address?
If that's okay, yes, I will share
Yes.
it in the
Yes.
show notes.
Yes.
Okay, Priya, well, thank you for coming on the
Thank
call.
you.
I appreciate it.
Thanks a lot for your invitation.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.